This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 469, recorded on November 24th, 2017. This episode is brought to you by the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. The department is presently looking to recruit prospective graduate students to apply to the program by the December 1st deadline. For more information, visit bit.ly/micromssm. This episode is brought to you by the Virology and Gene Therapy PhD program at the Mayo Clinic Graduate School of Biomedical Sciences in Rochester, Minnesota. The renowned Mayo Clinic Virology and Gene Therapy program is currently accepting applications until December 1. If you want to be trained in the exciting fields of oncolytic viral therapy, gene therapy, vaccine creation, or basic virology, apply to the Virology and Gene Therapy program at the Mayo Clinic Graduate School of Biomedical Sciences. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today in New York City, Dickson Despommier.
Hello, Vincent.
Happy post.
Yes.
I was going to say transcription.
Postprandial.
Thanksgiving.
Post-transcriptional. That's right. Did you have a good one?
Yeah, it was fine.
I did too.
Here in New York, it's sunny and 10 degrees Celsius.
Correct.
It was really very nice
and
yesterday.
There is no
today.
clouds in the sky.
Is that right?
None.
There are no clouds.
I am looking out the window and I see none.
Very good. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And I do not see a nun outside my window, but I do see clear skies.
She wouldn't dare walk past your house.
It's also 50 Fahrenheit, 10 C here, and was just a lovely autumn day. Did a bunch of yard work this morning, and
nice.
Did you cut the leaves?
I mowed the leaves. Yeah, I blew the leaves out of the gutters.
Mulched.
Excuse me?
Mulching. If you cut the leaves, that's mulching.
Yes, exactly.
From southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's 47 degrees, sunny, and that's 8 Celsius.
A little chillier, eh?
Just a little bit.
You don't have any snow on the ground, do you?
No, we haven't had any snow stick at all.
How was your concert?
It was great.
Tell us about it.
Well, let's get our last guest in,
all right.
Oh, yeah.
Okay.
so that he may speak. That's from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
All right.
How was your concert, Rich?
My concert's still yet to come. We have 74 degrees Fahrenheit. That's 23 Celsius. Get this.
Perfect.
27% humidity.
Nice.
Wow. That's a dew point of 38 degrees.
That's lovely.
And it's brilliant sunshine. And the only bad thing is that, except for going out to fetch the paper today, I haven't been outside. This is crazy. I need to get out of this place.
Go for a walk,
Sounds
man.
Right.
I will. Right after talking to you guys. I'll talk to you guys first.
Okay. All right. Kathy, tell us about your concert.
Well, I'm in the University of Michigan Choral Union. It's UMS Choral Union, not University of Michigan, but it is connected with the university. And we sang last Sunday with the New York Philharmonic. This is the 100th
That's exciting.
anniversary of Leonard Bernstein's birth, I guess, must be. And so we did the Kaddish Symphony, which is Symphony number 3, very seldom done. In fact, none of us had ever done it before. That never happens when he asks who's done this before. Usually there's some hands that go up. We worked really hard on it. It had a children's choir from Michigan State, and narrator was Jeremy Irons. And then there was a soprano soloist who was very good. And it had been done about 10 days beforehand in New York City, but this was the Ann Arbor version of it. So
how
it was
big
really
a choral group are you?
When we're full forces, it's about 160, 170.
Wow, that's fantastic.
So, yeah, our next big gig is the entire Messiah, which we do 2 performances of, December 2nd and 3rd.
I have one follow-up from Raihan, who writes, Dear TWiV hosts, I'd like to throw my name in the hat for the Viruses book giveaway, but more importantly, I feel like adding to the spin-off conversation you had about publishing and career advancement. It's clear from episode 468 that Dr. Amy Rosenfeld is a fully qualified scientist who knows the A to Z of virology, especially Zika. Being trained by Earth's virology professor clearly has made her an excellent scientist. It is also evident from her pick that she is someone with immense compassion. We often forget the purpose of doing research, that is to alleviate world suffering. Well, at least it's one of the purposes. I feel scientists have great propensity and opportunity to create change to this very dark world we are living in. Many of us got into science because we believed we could do that. But the pressures of publishing and reaching KPIs, key performance indicators, sometimes grind away at that initial drive and just make us into impact factor-seeking drones. It's obvious that Dr. Rosenfeld will make an amazing PI. It saddens me that someone like her has difficulty in getting such a position. I was taught by my PhD supervisor not to seek high-end journals. He was of the opinion that it's important, just important to get your science out. I agree with him, but sadly most scientists don't. I once met a young assistant professor who told me, you need to learn to play the impact factor game. He had several Nature papers, a company, and a faculty position. His words affected me quite badly, not just because I didn't have papers in high-impact factor journals, but because that is the sad reality. No one will give you a job because you're passionate or because you want to inch towards the solving of world's problems. I'm still proud of my work, despite it being very meager compared to other scientists of my age. If you check my Google Scholar page, please don't, but if you did, you'll see that my papers are hardly cited. In fact, the work that I'm most proud of was published in 2016 and has not been cited at all. But I still feel a rush each time I read and learn something new. I get all tingly inside when the data fit a nice curve or when a trend appears to prove or disprove a hypothesis. I do feel like this is the best career for me, but I just don't know how to play the game. Maybe I'm not a brilliant scientist. Maybe I'm delusional in thinking that someone like me can run my own lab. But does that mean that someone like me needs to live contract to contract, paycheck to paycheck? This industry is tough. There are too few permanent jobs out there, and I really hope not to still be a postdoc when I'm in my mid-40s. I wish Dr. Rosenfeld all the best in her research and her career. I'd like her to know that well-meaning and sincere scientists like myself are totally behind her. I wouldn't give the same play-the-game advice that I was given. I'd tell her, just keep at it. Don't let your drive and passion be replaced by wanting to seek publishing success. Let the science speak for itself and let your passion shine, and maybe, just maybe, but hopefully she'll be the one to break the mold. Many of us are trying. I know I am. Yeah,
it's interesting to comment that she's got 2 papers in PNAS and she doesn't have a job. So just playing that game at that level is not enough, unfortunately.
The latest thing now is that many programs give preference to applicants who are already funded.
Oh, gee, who wouldn't have guessed that one?
Oh, my gosh, it's so hard.
It's about overhead. It's incredible. It's incredible.
It's very tough.
Did you realize that I was a postdoc at Rockefeller when they didn't allow them to apply to NIH? They were funding all the research from within.
Is that right? They didn't allow it?
They didn't allow it. And then they started to allow it because they realized that they couldn't continue to do that. But for many, many, many years, they funded all the research. You didn't need a grant. That's incredible.
Raihan doesn't say what he's doing.
Right.
He's told us
industry,
whether he's a postdoc or he's in an industrial job or whatever.
Well, it seems to me that he's a postdoc, right? Because he said, "I don't want to keep being a postdoc till I'm 45." But he was originally doing Flu B, I think. Was this Raihan who did Flu B and wrote that wonderful
in plants?
Oh, maybe.
And then he wrote recently. I got it, TWiV 442, and I can tell you where he was. Raihan. Here we go. He's moved to another post. He's left Flu B because I remember he wrote, I left the love of my career, Flu B. I don't know where he is, but I suspect he's doing another postdoc.
Well, I mean,
right.
The only thing I can say is try not to beat yourself up so much, I mean, clearly he loves science, and that's great. And I hope he can somehow parlay that into a stable living. And I'll be interested to find out how that plays out. But beyond that, and I'm as guilty as anybody, but spending time comparing yourself to other people and beating yourself up about how much success, in quotes, that you've had is it's tough, I don't know how, you can't just say don't do it because everybody does that. But
yeah, I think everyone compares, and I always tell people don't compare yourself to other people. You can't really make it. But
unfortunately, when you apply for tenure or put up for tenure, they demand that you come up with two lists.
Of course.
One is the people that will support it, and the others are the ones that you should be compared to. And you can't get around that.
I saw the tenure, the generic letter that was sent out for my tenure, and it said, please, compare Dr. Racaniello to all these people, and if any of them are better, let us know. We'd like to hire them. Isn't that nice?
Yeah.
And it's from the minute you have any consciousness, you're being evaluated and compared, right? You get grades in school. You're told what's good, what's bad. A lot of this stuff is part of becoming civilized, but it's overcooked, and you can get yourself into a situation where you're just beating yourself up all the time, and you rob yourself of the enjoyment you can get from your passions. So
at what point do you decide that you can't have your passion?
No, I don't think the passion goes away, but
no, you can't have the job that's fueling your passion, right? I mean, you cannot try forever, right?
Yeah.
At some point, you have to say.
Well, you could, but at some point, it'll become untenable.
Yeah.
I don't know. I don't know the answer to that.
There isn't an answer, but everyone has to feel at some point, what should I do now? I've tried this for so many years and it's not working. What do I do?
We should all have a plan B.
I think so.
Yeah.
Sure.
I think so. Yeah.
Well, this is an ongoing conversation here, and we're going to, depending on how this episode goes, we may get into a little more of this. And I would really like to hear from other listeners about, what their life is like. How. Maybe some who have decided, no, I can't do this anymore. I'm going to do something else. Whatever.
Yeah, this is something that I added to my careers talk, a little introduction where I talk briefly about, the inherent bias of these types of discussions where you hear from the success stories. There's the survivorship bias built in, and nobody convenes a panel of disgruntled former postdocs. But it would actually be kind of useful if they did. Like, when did you decide to move on to something else? What did you decide to move on to?
Hey, they're running our journals right now.
Some of them are, but then there are
screening
also
grants at NIH.
There are probably people who are doing something that we hadn't thought of that might be quite interesting, or
right, and who are very happy with it and are glad they finally jumped ship.
Right, and whether they're happy with it or not, I think it would be a perspective that would be useful to get. So, attention, disgruntled former postdocs, write to us.
Yeah.
All right. Thank you, Raihan. This episode is brought to you by the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. They want you to know about their graduate and postdoctoral programs. Composed of over 20 virology labs, all centralized in one building in the heart of New York City, this department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to the program by the December 1st deadline. Interested postdocs are also encouraged to contact faculty of interest. For more information about the department, visit bit.ly/micro MSSM. And as a former graduate of that department. I can vouch for it as an excellent program. Let's see, they work on viruses, including influenza, hepatitis C, dengue, West Nile, HIV, Ebola, Marburg, Crimean Congo, hemorrhagic fever, cytomegalovirus, and others. Lots of different faculty: Peter Palese, Adolfo Garcia Sastre, Ana Sesma Fernandez, Matt Evans. Who else? Many more. I want to name a few more. I'm on the website, by the way. Find faculty.
We're not.
Find faculty.
We're just listening.
Now, our disciplines, our curriculum, our students. Where is our faculty? Why can't I click very quickly? I shouldn't really say this to a paying advertiser, but here we go. Here's some more faculty. Nicole Bouvier, Ben Chen, Matt Evans, and then I have to click Show More. Anna Fernandez Sesma, we mentioned. Florian Kramer, Ben Hurley, Gene Lim, who Rich knows from Florida. Show More. Why do I have to click Show More?
They want to track how many people keep scrolling.
You think?
Maybe.
I don't like it. Megan Shaw, Viviana Simon, Ben Tenhoover, of course. And this one I just want to mention because I want to pronounce it. Domenico Tortorella.
I'll have another helping of that, thanks.
Melissa Uccellini. I think Uccello.
Does Domenico Tortorella work on Torquetano virus?
Now listen, Melissa Uccellini. Uccello is little bird.
Oh.
Uccello. And he was also an Italian.
So Uccellini would be little bird?
Yeah, I think so. Let me make sure. What is an Uccello? It's a small cello? No.
No.
All right, I have to use the Italian translate. All right, so we'll put Uccello here, and of course it tells me that it's Uccello. No, come on. All right, so no, it's not. Pipistrelli is bat, right? So Uccello is a little bird. Yeah, I got that right. All right, so that's that faculty there. Enough of that ad. That was very good, though. And then we also are sponsored by the Mayo Clinic, the Virology and Gene Therapy PhD program at the Mayo Clinic Graduate School of Biomedical Sciences. They want all interested individuals to apply to their fantastic program. This program has the stated mission to train students to work together with scientists and physicians to address important questions in fundamental virology as well as more translational applications using viruses as tools for gene delivery or oncolysis. Students encounter small class sizes in courses taught by experts in the basic principles of virology and the translational application of viruses. This is a competitive graduate program with average time to degree of 5.2 years, with over 90% of individuals successfully pursuing the career of their choice after graduation. The graduate school offers a stipend of over $30,000 annually, which is...
Oh, good, because last week I heard you say 3,000.
Did I?
I may have misheard it. But
that would be bad.
Yeah.
Bad.
Cost of living in Rochester is probably reasonable, but not quite that reasonable.
30,000, and hopefully your tuition won't be taxed.
Yeah.
Right.
With special programs for additional financial assistance, especially for those from disadvantaged or underrepresented backgrounds. Through the unique funding mechanism of the school, students receive their stipend from the graduate school, not from a PI. Therefore, grad students are completely free agents, able to choose the lab and situation that is best for their careers, not based on extenuating financial factors. Students have no requirement to serve as teaching assistants. However, opportunities in entrepreneurship, communication, and teaching are available through career development internships in partnership with local businesses, publications, and undergraduate institutions. Mayo Clinic is in the city of Rochester, Minnesota, that boasts a low cost of living, the perfect environment for a graduate education. Apply and begin your graduate education in the frontier of translational virology. If you want to be trained in the exciting fields of oncolytic viral therapy, gene therapy, vaccine creation, or basic virology, apply by December 1st. You can find the application at mayoclinic.org and click on the education tab, or you can just Google virology and gene therapy, click on Mayo Clinic Virology and Gene Therapy for more details. All right, this is the last time we'll mention both of those programs, and December 1st is next Friday, also known as Kathy Spindler's birthday.
Yeah.
So that's two important deadlines, two important dates.
That's right.
All right, we have a paper for you, specially selected for those who celebrate Thanksgiving, because it involves viruses and fungi and plants. And if you think about it, you can eat a lot of plants on Thanksgiving, and if you keep the leftovers too long,
fungi
growing
no,
on.
you can
eat fungi.
I was
Lots
wondering
of them.
how
We
you
had
were going to get around to that.
wonderful fungi for Thanksgiving.
And of course, plenty of viruses.
Oh, yeah.
I thought you were going to turducken, that it's a virus inside a fungi inside a plant.
That could be. Viral turducken. That was the name of an episode, right?
Yeah, unfortunately, we already did that title.
Yep.
But,
Right.
that would be seasonal. And in fact, we're going to talk about a virus they isolated from inside a potato.
Potato, which I ate yesterday.
Yeah.
Probably several of you did. Did you have potatoes, Dickson?
Sure.
Sweet potato.
I had sweet and white.
Didn't have white.
I could do without the white, the little round roasted whites. I don't like those, but the sweet was great because we had put bourbon in. Actually, not bourbon, Jack Daniels.
Oh.
It's a bourbon.
Is it? It's not really, is it?
Jack Daniels is a
Sure.
bourbon. Of
Yeah,
course
that's
it's
a bourbon.
Well, we had a bottle of Jack and a bottle of Bullet on the counter. My wife
Bullets.
said, use the Jack because it's
Yeah, because
cheaper.
the bullets are cheap. That's right. That's true.
But it was fine. It tasted great. All right, the title of this paper, which was published in the Proceedings of the National Academy of Sciences, early edition, Phytopathogenic Fungus Hosts a Plant Virus, a Naturally Occurring Cross-Kingdom Viral Infection. And the first author is Ida Bagus Andika, and then we have Wei, Cao, Salipeth, Kondo, and Sun. And they're from Northwest A&F University in Yangling, China, Okayama University in Japan, the Inner Mongolia Academy of Agricultural and Animal Husbandry Sciences, which is in China, and King Mongkut's University of Technology in Bangkok, Thailand. Wow, what a diverse institutions,
like
right?
a pan-Asian sampling of scientists here.
And I must say, I have not heard of most of these institutions.
It would be interesting to know how this collaboration came together.
Would be.
Yeah.
Well, probably over a meal of potatoes. This was edited.
More likely rice.
Rice. This was edited by Jim Van Etten at the University of Nebraska-Lincoln, which is interesting because he's interested in unusual viruses.
Mm-hmm.
So they probably thought, let's send it to him. All right. So this is all about viruses that cross kingdoms. Now, Dickson, you know what a kingdom is, right?
I do.
Can you tell us the kingdoms?
Well, there's the kingdom of Thailand, and there's the kingdom. Oh no!
Thank you. No. Let Rich take it, Alan.
Me?
No, take it, Alan.
The problem is that this is a moving target,
This
right?
is true. This is all.
How do we define what's the modern? There's plants,
5
That's
of
right.
them now.
There's animals. Right.
there's
Archaea,
archaea, what else?
Bacteria.
Eukaryota.
No,
no.
protists and fungi.
Protist. That's
Protist
protist
and
and
fungi.
fungi. That's right. 5.
Right,
So
the
there's
thing-
actually 7.
There's
Wait,
7
on-
kingdoms?
1, 2, 3, 4, 5. No, that's 6. Well, I went to Wikipedia, and there's a big debate. There's a
There's
lot.
always
that's
a big
that's the
debate.
problem. Like I said, it's a moving target.
Well, the thing is, we've got 3 domains of life.
Aha.
That
That's
it
why it gets
used
confusing.
to be. It used
that's
to
it,
be that
that's it.
kingdoms were the top level classification, but then
Domain.
it became clear that that we actually had these 3 major domains. You have the archaea, the prokaryotes, and the eukaryotes.
Right.
Okay.
And then below that, well, where do you put things? I mean, plants and animals are both eukaryotes, so they're both in the same domain. Fungi are in that domain. These used to be kingdoms. But then archaea, they're their own domain. So are they also a kingdom?
Shows you how silly it all is. It's all silly.
Right. It's a bigger version of the species problem. You know, what defines
Correct.
a species?
That's
And
right.
now you get all the way up to the top level and you say, well, what's the top level classification? And the domains make sense, but then kingdoms get a little fuzzier.
They do.
It's important to keep in mind that all of this is made up.
Yes.
Right?
Correct.
Okay.
They vote on this. They vote
in my
on
mind,
this.
at least, nature is
That's right.
essentially a continuum.
No kidding.
All right.
That's correct.
And we divide it up to try and make sense of it for ourselves, to categorize things.
Yeah.
Okay?
So you're
all
actually
made up.
denigrating this paper, Richard, by saying that, because they want to say that this virus has crossed kingdoms. The first example, come on.
But there is some basis. It's made up, but it's not made up just for the heck of it.
No.
It's made up to try and organize things.
There are rules.
And there are rules, and you can say that there are distinct features of a fungus that distinguish it from a plant.
That's correct.
Right.
It's not ambiguous.
And it's fairly clear at the top level what's a fungus and what's a plant.
Exactly.
And so, it seems like there ought to be some kind of distinction between those things. And so we can say, all right, they're different kingdoms. They're really, really different things. It gets across the point that these are biologically entities that you wouldn't expect to share stuff like a lot of genes or viruses.
Yeah, but when you just say we shouldn't expect, that requires an opinion about this stuff. Why should you have an opinion? Just go with the data.
Yes, you should, but it's useful. I think it's useful to be able to categorize things and say, all right, here's the kind of stuff we might expect a fungus to do. Here's the kind of stuff we might expect a plant to do. And then when we find data that go against that, we say, hey, that's interesting. Let's get a paper in PNAS because we've got something that's...
Or someone else says, that's crap. That can't happen. It's absolutely against all the rules that I've set down in my paper.
Right.
Well, the interesting thing about this is that, as Alan said, there's really clear distinctions between some of the things that we have identified as being in different kingdoms, like animals and plants and fungi. And the interesting thing is, and there are differences even at the molecular level, intracellularly, how things work.
Of course.
And so it's interesting that viruses can move around from one kingdom to another. As a matter of fact, you find what we would call families of viruses like reoviruses or rhabdoviruses that have all of the hallmarks of an animal rheo or rhabdovirus that infect are living in other kingdoms, indicating that what we think of as virus families have evolved kind of independently or no, probably by this kind of stuff, moving around among kingdoms.
Move around. So there are some examples of viruses that infect members of different kingdoms, and typically an insect is involved in bringing it from one to another. There's a recent report on a fungal virus that infects an insect and is transmitted that way. And you can do cross-kingdom infections in the laboratory. Don't hit the table every time you get excited.
I'm getting excited now because if you just happen to think about the virus that we've had a lot of discussion about, West Nile virus, we call it promiscuous.
I don't call it promiscuous.
Well, some people do. And they do that because it infects so many different things. It infects insects, it infects cold-blooded vertebrates, it infects warm-blooded vertebrates, it infects birds, it infects mammals, it infects... What doesn't it infect?
Well,
it doesn't infect fungi, probably, and it's also transmitted by a mosquito, so it also infects the mosquitoes. And is it the main host of that virus, or is
it's not different kingdoms, right?
Well, no,
that's what we're talking about here.
These are animal kingdoms, but look at the range just among the animals.
Some viruses have a broad host range, some are narrow,
and some are extremely narrow.
Cross kingdom is really pushing it because we don't know many examples of that.
Because you haven't looked at plants much.
Yeah, sure. That's definitely
angiosperms. Remember, those are flowering plants, and we surprised you.
Sure, I bet if you look more, you'll find more. But in the lab, you can do cross-kingdom infections. The one that I think is best known is from Paul Alquist's laboratory, who got a plant virus to replicate in yeast. It uses that as an experimental tool, right? And there are other examples as well. So in this paper,
this one you picked out, Vincent, that they refer to, the insect virus infecting the fungus. I think that's a paper that we ought to stick in the queue. I looked through that, and that's
cool, isn't it?
every bit as cool as this paper.
Yeah, I agree. It's 2016, and I didn't see it, but we'll do that one. Yeah, I agree. So this paper, they want to know can they? Well, they found it, and now they're backtracking, saying But this is about fungi, and they make the statement, which I wanted to run by Dickson. Virtually all flowering plants are exposed to attack by pathogenic fungi. They're called necrotrophic pathogens, which kill and absorb nutrients from dead host tissue. Then they have
They eat the dead.
They eat the dead.
do.
Biotrophic and hemibiotropic, which colonize the host and develop ways to take up nutrients,
Right.
including injecting effector proteins into the plant.
Oh, yeah.
So they have the capability to put things in, which immediately brings to mind viruses, right? It's also been thought that little RNAs can be transferred from fungi to other microbes or even to plants and so forth. So the idea being that there could be exchange between fungi and plants.
Right.
There are many viruses that infect fungi, mycoviruses, which I think we've talked about here, including, I think we talked about one that reduces the virulence, viruses that reduce the virulence of their fungal hosts, right? But they say no contemporary cross-infection of a plant virus to a phytopathogenic fungal host has been described. And that's what they're going to do in this paper. And the fungus that they use is Rhizoctonia solani, which we'll refer to as RS. It's a soil-borne plant pathogenic fungus with a wide host range. Dickson, field crops, vegetables, ornamental plants, alfalfa, shrubs, fruit trees. Man, you think West Nile is something. Look at that.
No, I'm impressed. Very impressed. And they call it cucumber mosaic virus.
Well,
and then they refer to it as CMV, and that's very confusing.
Yeah, that's confusing.
Cytomegalovirus, yeah.
Right.
Well, there are only so many letters. Dickson.
This is all true.
Diseases caused by this fungus include root or stem rot, stem canker, and damping off of seedlings. It is a basidiomycete, does not produce asexual spores, which are also called conidia, but it exists as a vegetative mycelium.
Can you name another basidiomycete, Vincent? Name one.
Ascaris lumbricoides.
No, that's a North Korean disease.
I don't know. What's another basidiomycete?
A field mushroom.
And I'm not looking it up.
They're
See,
all
I'm being
mushrooms.
good.
They're all mushrooms.
A mushroom is a basidiomycete.
Absolutely.
Ammonidis phalloides. Ammonidin phalloides.
See?
Yeah.
You could name one.
Yeah, but I didn't know. I didn't realize because I don't think about fungi enough. But we should.
Well,
because they're cool.
They're very cool.
My son told me the other day.
They taste
They're
good.
the world's largest organism.
This is cool. My son went to the supermarket and he bought mushrooms.
Oh, yeah.
He said, I put it in the fridge, and the next day they were covered with fungus. It
No, they were covered with slime mold.
was all fuzzy white stuff.
Yeah, that's right. That's right.
Isn't that funny? A
It
fungus
is.
growing on a fungus.
Well, that's the way it goes.
But the white one would not be a basidiomycete,
Yes,
right?
it would. Yes,
It would be?
Yeah, sure.
He probably could have eaten it, right?
Field mushrooms are that's the commonest mushroom, basically.
So the other thing we need to know is R. solani is known to harbor mycoviruses. Okay. And they say something that's really cool here, including some that do not belong to any established virus families or genera.
We don't know where they're
So
from.
These are
Came from outer space.
no, what do you call when you don't want to be part of anything? You're
Misanthropic.
no, there's another word. They want to be
Isolationists.
what's the word? Oh, I can't think of words anymore. I think I have to stop TWiVing. I can't
Are they rogue viruses?
rogue viruses.
Rogue.
They're rogue viruses. Okay. And now the other virus here
Hermit
is
virus.
in this paper, as you will see, they discovered that cucumber mosaic virus can,
Which we ordinarily think of as a plant virus, cucumber
Indeed,
mosaic.
indeed.
They found
Cucumber mosaic virus, though, is also a broad host range.
it is
pathogen.
the largest. They say it has the largest host range of any plant virus,
Goodness.
which I would say known.
And they all cause mosaics in those plants? That is, the leaves have different colors?
I hate to say all, Dickson. I just don't know.
Okay.
All right, so they found that a form of cucumber mosaic virus was found in a R. solani strain isolated from potatoes.
And what kind of virus is that, by the way? RNA?
Plus stranded, single-stranded
Plus-
RNA.
Excellent.
Single piece of RNA.
Cool.
No, actually, it's
sorry,
it's three pieces of RNA.
3 pieces. Just making sure you're listening because it was pretty quiet there.
And it's in a naked capsid.
3 naked capsids.
Naked capsid.
Yes.
And if I understand it correctly, each RNA is encapsulated separately.
That's right. Exactly.
Wow.
Separately. And you need all 3, right, to get an infection?
Mm-hmm.
I think so.
3-hit kinetics.
Mm-hmm.
So CMV is vectored by aphids mostly.
Aphids.
Yeah, it's right here.
Mm-hmm.
And, okay, so first of all, they were interested in viruses that affect this fungus, R. solani. So they screened fungal isolates from potato plants grown in the Inner Mongolia province of China.
Wow.
So that's one of the sources of the collaboration, right?
Right.
Mm-hmm.
They look for double-stranded RNA.
Right. And I had a question about how they did this. So I tried to track it down in their methods, which were in the supplemental section, and that referred to another paper, and then that kind of referred to another paper, and I was never satisfied that they really were ever going to explain it. And Rich and Vincent came up with a method that has been used, but it's not clear that it's what these authors used to isolate their double-stranded RNA, and Rich can tell about it.
It was in the pre-show.
It's what I would use. It took me back. I can visualize myself at the bench during my first postdoc.
We were talking about it in the green room. Excuse me.
That's another way to put it, yeah. This is the green room. Yeah.
It's called a Franklin column, named after the guy who published about it, Franklin. I even found a paper. It's a 1966 PNAS paper where he's separating replicative intermediates of bacteriophage R17 RNA. And all it is Wattman's CF11 cellulose. And I had the method right. Can you believe this? This is almost 40 years. You make RNA and dissolve it equilibrate the column in a buffer containing 35% ethanol, and pour the RNA in the same buffer over the column, and it all sticks. Then you reduce the ethanol to 15%, and the single-stranded RNA comes off. And then you just dilute it with no ethanol, and the double-stranded RNA comes off. And so it's as clean as a whistle.
You stick it on a big stack of paper.
Yeah.
And then you can you can
just
peel off.
Plain old cellulose.
It's New York Times.
I mean, like, why?
So, you could wrap fish in it, or you could isolate double-stranded RNA.
Ooh. If you wrap 2 fish in it and give it to a not-so-good friend.
Oh, yeah.
That's curtains. Can I raise a side issue here? What was the cause of the potato famine, by the way, the Irish potato famine?
It was a fungus, but
that's what I thought.
Sort of blight, right?
But I wondered if they actually proved that it was the fungus or a fungus plus a virus. I don't know. I don't know.
It's an interesting question, Dickson.
I just raised it as a
sharp thinking.
Well, not necessarily.
Now, of course, the Irish potato famine. You get a whole bunch of history stuff about how it wasn't actually the death of the potatoes that was causing people to starve. It was the economic conditions at the time.
It was the English that did that.
It was
yes.
Potato blight. It's an amoeba protozoan.
Really? It's like it's a like Dictyostelium.
Not touching it. Not touching it.
Phytophthora
infestans.
It's a new kingdom that we've never.
No, and they used to think it was strain US1 that caused the blight, but it's now known to be Herb1.
Interesting.
Which had originated in the Americas, most likely opposed to Toluca Valley.
Right.
Sometime in the early 19th century before spreading to Europe in the 1840s.
Bingo.
So they looked for double-stranded RNA as a marker of virus infection. So of these 29 fungal R. solani strains, they found 11 that had double-stranded RNA in it. They picked 7 for further analysis, and they showed that they eventually did some deep sequencing of RNA, and they found, mycoviruses, but they also found something which was very similar to the 3 RNAs of cucumber mosaic virus.
So a couple of points here. First of all, that you would, the reason for looking for double-stranded RNA is that most fungal viruses are double-stranded RNA viruses.
Mm-hmm.
And you wouldn't go looking necessarily for a single-stranded RNA virus, not that they were, by looking for double-stranded RNA. However, in order to replicate, there's got to be double-stranded RNA replicative intermediates, and that's how they found this thing.
That's right.
The other thing that I think is really important that they emphasize about this is that this came off of a fungus from potatoes growing in the field. So this is a naturally occurring infection. This is not something they found this out there.
That's
why I went to the Irish
potato
famine. That's
I mean, way out there. We're talking Mongolia, right?
Okay.
Well, you got an inner and outer.
This was inner.
This was inner Mongolia.
And by the way, remember where potatoes came from originally?
Where?
Peru.
And where did pasta come from?
It came from China.
And pizza?
Don't know.
Okay. You're the limit of your culinary knowledge.
Limited at pizza.
Anyway, this virus that looked like CMV, it's 3 RNAs, typical organization, coding, and so forth.
Mm-hmm.
And so then they went back to one of the fungal isolates, and they did some RT-PCR and sequence analysis and showed that it was in there. And then there's one, they have a cured strain.
Aha.
Does anyone recall how they cured it?
They didn't say. Did you look it up, Kathy?
I didn't look that up. I just took their word for it.
Yeah, they say not the cured strain. All right, so all of a sudden they throw this at you. Cured? But you just discovered it. How could you cure it? So they don't tell us. But anyway,
I think it grew from the basidiospores because it didn't infect the basidiospores.
That's probably how they cured it. Very good, Dickson. Awesome.
Lab
really
science.
Good. Really
Lab
good.
Science.
But you're thinking spores. I like that.
I am.
Anyway, so they cure it, and the RNA is no longer there. There are double-stranded RNAs in these CMV-infected fungi, so that would explain how they originally pulled it out.
Mm-hmm.
They have the sequences show the mycoviruses as well.
Mm-hmm.
All right. Then this is a cool experiment to validate that it's the CMV. They call CMV-RS for the fungal name RS. They take total RNA extracted from a strain and they inoculate it into the leaves of Nicotiana. Kathy Spindler, Nicotiana. Did I get it right? Did I get it right?
Yeah. Yeah.
A tobacco-like plant?
I always say it wrong.
Yep.
No.
But
it's an Australian plant.
Yeah, no, I know that plant because
so they rub inoculate, mechanical rub inoculation.
That's the one they used in to grow the influenza virus in Denver,
polio
Texas.
Virus, yeah, polio too, yeah.
That's right.
We did a polio vaccine paper, right?
Yeah.
And that's when I mispronounced Nicotiana.
Highly susceptible.
7 days after inoculation, the plants show a typical mosaic symptom. Cucumber mosaic virus. Now Dickson is say, wait a minute, if it's called cucumber, why does it infect Nicotiana? Are you going to ask that?
No.
No, good.
Because we already discussed that it's promiscuous.
Good.
That's right.
And they found virus in the upper leaves by RT-PCR. So in other words, the total RNA from this CMV-infected fungus can cause what looks like a CMV infection in a plant.
Here.
Nicotiana. That's a cool experiment, isn't it?
Yep.
Now, is this the one where they also show that there's no evidence of the fungus up in the leaves? Or is that a later one?
That's later. That's cool, though. That's
sorry.
very cool. It is later.
Excuse me for just one moment.
Where are you going?
Can I just this is really off the wall here.
It's okay.
But I have an in-studio guest that wants to say hi.
Oh, hi, Harper.
Hi.
How you doing, Harper?
Harper.
I got your name this time.
Yay.
Yeah.
And remember, Harper is listening.
Someone on a recent TWiV, their pick of the week was To Kill a Mockingbird.
By Harper Lee.
Yeah. And I thought of you, Harper. How about that?
Yay.
Did you have a good Thanksgiving?
Yes, I did. It was really fun.
Harper, you would never kill a mockingbird, would you?
No, I wouldn't.
I didn't think so.
Would you mock a killing bird?
Oof.
No.
Oof.
Oh, okay.
Do you know what a mockingbird looks like?
I don't think so.
Yeah.
We have them up around here. They are called mockingbirds because they can imitate the voices of other birds.
Mm-hmm.
Or anything.
Or
there
anything.
were some in New York that used to imitate car alarms.
True.
Are you serious?
Yeah.
Yeah.
Oh my gosh, that's amazing.
I bet you they could imitate Harper if they wanted to.
Yeah.
All right, guys.
How old are you, Harper?
I'm 9 years old.
9 years old. You are articulate. Your pronunciation is impeccable.
Very good.
Did you know that?
Because of that, you should, do our closing thing for us.
You could come on and read a few emails for us.
Or you could just get on tape saying another TWiV is viral.
Oh, that would be great.
Can you say another TWiV is viral? The silence.
Nothing. Harper, can you say another TWiV is viral?
No, she doesn't want to do it. Okay, we'll try next time.
Okay.
Next time. Bye, Harper.
She'll be in the audience in Austin.
Oh, cool.
All right.
Oh, I'll bet she'll come up on stage and do it, right?
Could be.
By then you'll be older.
Exactly.
Nice to see you, Harper.
Or hear you.
I'm gonna go eat lunch.
Do that.
Are you gonna have
what's for lunch?
Turkey?
I'm gonna have chili.
Chili.
It sounds good.
Sounds good.
It is Texas.
Enjoy.
There you go.
Thank you.
Bye-bye.
Bye.
All right, bye.
Sounds adorable.
And then they wanted to know if this CMV RS could infect other. Wait, we questioned whether R. solani, could host other CMV isolates. Again,
right.
I'm mixing up my experiment.
Right.
So they have an isolate which is 95% identity, was introduced into a virus-free or a double-stranded RNA-free strain of RS by transfection of fungal protoplasts with purified virus particles. It's very interesting. Kathy, do you know what kind of transfection conditions they use for that?
Electroporation.
Is it
to transfect protoplasts?
with virus particles? Yeah. Is it chemical transfection? I would assume.
I don't know.
Yeah.
It might be electroporation. I don't know.
So these are virus particles, CMV virus particles. Okay. And to get them into the fungus, they transfect them because there's no receptor on the protoplast,
Exactly.
right? So they transfect them in, and then they can see whether it's producing proteins and replicating. So they could see accumulation by RNA or Western blotting. And so this R. solani can support the replication of a different CMV isolate. And furthermore, the virus is stably maintained in successive cultures. Of course, means not killing the fungus, right? It's replicating in it, but not killing it. And did not affect the morphology.
Also interesting.
And this, I'm saying just because I love the name of this medium, potato dextrose agar, PDA. I love that name. I've heard it before. All right. Now, I think we mentioned before, mycoviruses are typically transmitted horizontally via hyphal anastomosis. Yes.
That's like fusion, right?
No.
Correct.
Have you ever had an anastomosis, Dickson?
Only when I was embryologically developing.
Because some people at older ages, they can have their intestines anastomosed.
Yeah,
right?
I haven't done that.
You can have a piece cut out. And
No, I'm working up to that one.
Or
Alan Alda.
Did he have that?
Oh.
Yeah, and it happened when he was in South America, and when they started describing it to him, he knew what it was because he had done the surgery, so to speak, when he was a doctor on MASH.
That's funny. That's very funny. So horizontally via hyphal anastomosis. So in other words, two hyphae can join, and then the virus is transmitted. Because these viruses typically are not extracellular.
No.
These fungal viruses. Or vertically through sporulation. Right? So that means the virus would get into a spore as it's formed, and then as the spore germinates later on.
Right.
Oh, so that wouldn't be the way they cured it.
No.
Well, this one is not transmitted that way.
Aha.
Oh, it's not. Okay.
That's right, because they electroporated it into the
right.
fungus.
So it is not transmitted. It is transmitted by hyphal fusion, which you can easily do by just culturing two isolates.
But it's not
next to it.
transmitted vertically.
That's right.
Yeah, but it's not through spores.
So that's a very important process, by the way, in terms of linking vegetation in the wild. It's called mycorrhiza. And the mycorrhiza, they form these networks of fungi under the soil, which also connects the root tips together for all of the trees.
Right. So, Dickson, sometimes you can see a ring of fungi.
You can. A fairy ring.
Why are they in a ring?
Because the basidiomycete that forms in the center explodes in uniform direction. Unless there's a wind going when that happens.
I thought it was because of the mycelium underneath.
No, it's called a fairy circle.
Okay. But they are connected by mycelium.
They are. So that you can get fairy circles at the edge of the first circle And then at the next circle.
It's very cool, isn't it?
Next circle. Yeah. They're amazing animals.
Is that related to
Or
crop
plants,
circles,
rather.
Dixon?
No.
Okay.
That's
But they're
extraterrestrials.
They're called fairy circles because before people knew about the existence of any of this, they hypothesized that this was where fairies met to do their magic.
They blamed it on the wee people.
Yes.
So this virus, this CMV-RS, is transmitted via horizontally by hyphal fusion, but not via spores.
Right.
And that.
Right. So they could have cured it like that.
Yeah, that's how it worked, Dickson. And I remain impressed that you came up with that like this. At your age, you're still with it.
So,
yes,
pardon me, I was a little distracted. It does say in their procedures that CMV cured RA1 strain was obtained by single basidio-spore isolation.
There you go.
Bingo.
Confirmed. You no longer need to speculate.
Do I get my grant?
You have to write it first.
It will get you $30.
All right. So now, can this isolate CMV? Can CMV replicate in other fungi? So they took CMV FNY, which is the second isolate they looked at, and they looked at 3 phytopathogenic ascomycete fungi.
Right.
Valsamale, Cryphonectria parasitica, and Fusarium graminarium.
Fusarium, I'm familiar with.
You're familiar with these?
Yes, I am. I think that's wheat rust.
Wheat head blight disease.
And there's a Fusarium wilt, too, isn't there?
There is.
So the first one, Valsa, causes apple canker, which I love. Cryphonectria causes chestnut blight. And then Fusarium causes the wheat head blight.
Yep.
And,
it's a biggie.
I love these names and these words.
Yes.
I just love them.
They're big deals, though. If you want to eat, these are big deals.
So they could infect V. mali with CMV. It was replicating and it was stably maintained and transmitted horizontally via hyphal anastomosis.
Right.
And interestingly, the infected V. mali developed more aerial hyphae. I'm wondering if that is kind of a virus-induced thing, so it spreads more effectively to others. Because
sound like you're purposefully.
I'm not. I know it's no good to be purpose, but maybe a consequence would be more efficient.
Right.
And the virus-infected VM now, Valsamale, produced bigger lesions. And it is in a virulence assay on apple fruits and apple twigs. I don't think I've ever seen the word twig in a virus paper, but it's cool.
Have now.
So they have a virulence assay where they put the fungus on fruits and twigs, and they can measure lesions.
Right.
And having the virus in this Valsamale causes bigger lesions than not having the virus, right? So it's hypervirulence.
There you go.
The other two fungi were not good hosts for the CMV.
Right.
So that's cool. All right, the next question is, can the virus be infected from transferred from a plant to the fungus during infection in the natural environment? All right. But of course, they have to first look at this in the laboratory, which is not a natural environment, as we know, neither for plants or fungi or humans. So they did an experiment where they infect potato plants and Nicotiana plants with this CMV FNY. So they have the plant infect with the virus, and then they inoculate them with the fungus in the lower stem. All right? They let the fungus colonize the stem, and then they re-isolate the fungus, grow it, and ask, is the virus in the fungus? Got it?
Yeah.
Okay.
Right? Plants put virus first, then fungus, then take the fungus out.
Yep. Like a mixing chamber.
Right. So what they find is that the virus-induced fungus makes more severe stem rot. Sorry, the fungus makes more severe stem rot in virus-infected than in virus-free potato and Nicotiana.
A viral like Wolbachia.
So they say this shows that the virus infection enhances plant susceptibility. Then at 14 or 4 days after fungal inoculation, the stems are heavily rotted, and this is called damping off. So now I know what damping off means. They're heavily rotted, and the plants were collapsed, while virus-free plants retained vigor. And then they did RT-PCR and found that 38% and 28% of the fungi from virus-infected potato and Nicotiana plants were carrying the virus, CMV FNY. So in an infection of a plant, the fungus can get virus in the plant in a laboratory. That's a pretty cool experiment.
That means it's probably happening outside too, then.
Maybe.
I mean, there's a lot of outbreaks of plant diseases that I follow just by looking at ProMed. And you wonder, who cares about this stuff, right? And then
people do care. Yeah.
Everybody should care because those represent food sources for lots of people.
Sure.
And that
huge food sources.
Not available anymore.
And let's bear in mind that they isolated this virus from potato plants growing in the field.
Exactly.
So this, you could say that they replicated this in the laboratory, but you also kind of have evidence of the endpoint of it coming from the field. So it's not absolutely proven that the virus is going from the plant to the fungus under normal conditions, but it sure looks like that's going on.
Exactly.
So then the other question is, can the virus go from the fungus to the plant?
Right.
So here we've shown it go from the plant to the fungus. If you inoculate a plant with virus-infected fungus, will you get free virus in the plant? Okay. And so that's what they did. They put the virus-infected fungus in the plant, potato or Nicotiana, and then they look at virus accumulation in the upper leaves. So they put the fungus in the bottom stem, and they look for virus in the upper leaves. Stem rot disease was induced by this fungal infection. Okay, but virus-free fungus caused only mild stem rot.
Consistent with their previous results.
Right. So when you have the virus,
are worse.
They're worse than the fungus alone.
What about the virus?
No obvious virus mosaic symptoms.
Okay.
Virus was detected in the upper leaves, but no mosaics.
Huh.
And this is what Kathy referred to before. No fungus in the upper leaves. Right, Kathy?
Right. So the virus got there on its own.
Yeah, got virus. Apparently was
pathology
released.
is a synergism.
Yeah,
for mosaic.
Not for mosaic, for the stem rot, the fungus-induced disease. That's worse when the virus is present.
But you needed both for the mosaic.
There's no mosaic in this experiment. There's no mosaic, right? Which is funny because the virus is accumulating in the upper leaves, but for some reason not causing a mosaic. But if you do inoculate plants separately with virus, you rub the virus on the leaves, you do get a mosaic, but not here. I don't know why.
Location.
So this shows that the virus can go.
I wonder why they didn't use Arabidopsis for their laboratory model. I know it's not an edible plant, but it's a very common laboratory model where the genetics is so well understood that you could start to look into susceptibility factors and stuff like that.
Don't know, Dickson.
It's also a very small plant.
It's a very small plant.
And they may have wanted something that was a little bit larger so they could do this inoculation in one place and analysis from another part of it. It's just a guess. I have no idea.
Our most recent paper, plant virus, I think they did use Arabidopsis, and it is infected by CMV. Okay. So maybe it's something else. Okay, so those are the data.
Interesting.
And so they say, to our knowledge, natural infection of a plant virus in a fungus has not been reported previously.
Right.
All right. So this is pretty cool stuff.
So you mentioned the word ascosphere. You study, you've mentioned
Basidiomycetes and ascomycetes.
And so ascomycetes. People out there might not be familiar with what an ascomycete is.
Neurospora crassa.
Of course. Or Penicillium notatum. And
what's the difference? Tell us.
Well, the big difference is, just between those two fungi, no, the ascospores are definitely the same, basically. And there are 8 spores inside.
In a
and the genetics.
Ascomycete.
Yes, and the base, I knew Tatum, by the way.
Yeah, and Basidiomycetes make fruiting bodies, right?
That's right. And so the genetic organism of Drosophila was in conflict with the genetic organism of Penicillium notatum because they were in adjacent laboratories, and the banana media was being invaded by the spores from the ascospores. And
was this in Morgan's lab?
No, this is at Rockefeller. And they had to separate. They put them in a different building, so they didn't have to work together.
I have some answer to the question about the spheroplast transformation. Since you asked, okay, so it was reference number 3 in the supplemental data, which took me to a Journal of Virology paper, which then referred to another paper, which now I can't remember what the path was, but then there was a paper that no matter how I put the authors into PubMed, didn't seem to be indexed in PubMed. So then I had to go to the UM library and get the journal and download it. And it seems to be chemical transformation.
Chemical.
Is the bottom line.
Dear.
But that's why sometimes I don't go down those rabbit holes.
Interesting. There was a time when they used to have to transfect nematodes using a gun. Sort of like they propelled them so fast that it went across the cuticle. I don't think they do that way anymore, but I think at that point they didn't have any vectors. And so they actually had to use it. And I just thought maybe the fungus was like a
gene gun, but
exactly right. Exactly.
Chemical.
Right. Yeah. Okay.
So an interesting point they bring up in the discussion. So first of all, they say this is probably more widespread than we know, that these plant viruses can infect fungi, right?
No question.
Because otherwise, what were the odds that in the 9 plants that you pulled out of a field in Inner Mongolia that you happened to come across the one that was doing this?
Yeah,
that's it.
And they note that this fungus, Arsolani, is necrotrophic. It decomposes host cells, so that would allow flow of material like virus particles, right? So that would be an interesting way how the virus could go from the fungus to the plant.
Mm-hmm.
And the other point that they raised, which I think is interesting, well, 2 more things. First, that the virus makes worse disease. So that's important in terms of crops, right? You have to be aware of that. And the other is that the infection of a fungus would expand the reservoir of plant viruses. So now you have more viruses out there which could then go back to plants. So that could be a problem.
Right.
So you need to keep, I guess this is nothing stupendous, but you got to keep fungi out of fields, but that's hard to do.
There's
right?
no possible way that.
But if you did it in a vertical farm,
now you're talking. You're not just.
There's no dirt.
You're not. Besides, you can do barrier medicine basically, and you can keep out all the pathogens, and that's the advantage that I've been pushing. And finally, people are starting to pay attention to that. Actually, I'm not kidding.
Mm-hmm.
Because if you go to the Middle East, for instance, they've got wheat rust all over the place.
Yeah.
And then you go to Southeast Asia, you got rice blast. And these are killing off entire crops for lots of people. And the only approach now would be to keep your plants, watch out for these plant diseases by keeping your plants away from them. Because otherwise you're subjected to the whims of nature.
Well, clearly we need to do more on this.
We do.
These viruses, fungal plant virus exchanges,
Absolutely.
I think.
Absolutely.
So who would support, in the U.S., who would support
USDA.
USDA?
Yeah, sure.
So there you go. If you're a virologist and you're fretting about,
you know who else is really
animal viruses,
like all the agrochemical industries. They're major funders of this stuff. Monsanto, Cargill, Archer Daniels, Midland.
Right.
All those companies have deep interest in this.
I'm sure they probably have people working on it, right?
They do. Of course they do.
Oh, it's a secret.
Yes.
Well,
in the case of ADM and I think Cargill, it's particularly secret because they're privately held companies. They don't even report their finances.
That's right.
All right. Should we do a few email?
You bet.
Sure.
We'll probably have time to do 2 emails, I think.
2. Okay. Count them.
Because the second one is very long because it has a big, long response. But who knows? Who knows?
Read that one then. That's good.
Dickson, can you read that first one?
Sure. Mike writes, "Hello, TWiV. This email concerns an offhand comment made in episode 463.
Uh-oh.
Beware offhand comment.
That's right.
Actually, this is not bad.
While mega EV microscopes do exist, up to 3 MeV, they are generally used for materials sciences and physics rather than the biomolecular research for which cryo-EM is generally used.
That 463 was a cryo-EM episode, right?
Right. In the early days of TEM,
transmission electron.
Thank you, Rich. You can interject anytime you wish here. And you could be the translator. Increasing the accelerating voltage was a brute force solution to problems such as chromatic aberration, in which the electrons were not all emitted with a uniform energy/wavelength, and so were not focused to quite the same point. As the field matured, more direct solutions were found, for example, better sources with reduced EV spread, and the main reason for extreme voltages now is penetration through thicker samples. Structural biology has settled on 300 kV as the standard source for high-resolution cryo-EM. Looking at EMDB statistics. Okay, Rich, what does that stand for?
I got it. Electron Microscopy Data Bank.
Thank you.
Okay.
Thank you. All submitted structures marked as less than or greater than 300 kV seem to come from a single 400 kV microscope, which, incidentally, was destroyed by catastrophic flooding during Tropical Storm Allison in 2001. As an aside to that aside, lessons were learned from that flood, and the replacement microscopes remained dry during Harvey's recent deluge. The best, Mike W., and he's in Galveston.
So he ought to know.
He sure ought to know.
So I probably mentioned that. All right, so this episode was about the Nobel Prizes, one of them for cryo-EM,
that's right. That's
which one of the recipients is Joachim Frank here.
That's
so if it's German name, would you say Joachim or Joachim? How would you?
I think Joachim.
Joachim is
Joachim's
okay. And so he used to be at Albany, SUNY, or University of something up there in Albany. I don't remember where he was, but he was recruited here to Columbia, I don't know, 10, 12 years ago.
Interesting.
And my understanding was they had to give him a megavolt EM for his work. Okay. And he's got one. He's got one of the few. So I probably made the comment, the bigger the better. And so that's what it's about. Okay. But I think he does have a million electron vote.
I can remember there was a guy by the name of Riis, R-I-I-S, at the University of Wisconsin, that used a high-voltage electron microscope to actually look at living cells. And you could see them alive. They would not be killed by these extremely high voltages going through them. Mm-hmm. But they would be altered, I'm sure.
This is a pretty cool link.
Yeah.
This is EMDB. Nice. All right, the next one's from Mark. Dear Vincent and the TWiV gang, I was really happy when Rich brought up the hours worked versus productivity plot in John Udell's book on last week's TWiV. I'm a big fan of Udell and his ideas about science, but I'd seen that plot and it bothered me. First, the idea that you need to work insane hours to succeed in science is an assertion not backed up by data. And as scientists, shouldn't we be making data-driven claims? According to the plot, a scientist working 40 hours per week would produce about 2 millipapers per week, or 1 paper per decade. I get that the plot is meant somewhat facetiously, but that's pretty far off. Also, the productivity curve slopes increasingly upward as more hours are worked, but published studies support the opposite. As people work more and more, their marginal productivity actually decreases. To quote this article in The Economist, output at 70 hours of work differed little from output at 56 hours. That extra 14 hours was a waste of time. As it gives a link to an article called Proof That You Should Get a Life. So working more and more hours is like the plateau phase in PCR. You're doing more cycles, but you're not making more product.
Cool.
That's good. Second, as a group, we scientists can be obsessed with status, and bragging about how much you work can come across more as a kind of posturing to demonstrate your scientific worthiness than as a way to show the path to success. Why are you pointing at me?
Because I agree with that statement.
I never posture about how much I work.
No, but I've heard a lot of back.
I actually have never. I have rarely heard that. But I understand that
3 weekends in a row over. Come on.
You're hanging out with the wrong people. And I disagree with the. Wait a minute. Where did I stop? I, of course, agree that the more you work, the more you produce, but this is only true to a point, and I disagree with the implied notion that it's not possible to make reasonably valuable contributions to science while working relatively normal hours. Personally, I really love science, but I love other aspects of my life just as much. I think most scientists actually fall into this category, and that's why exceptionally productive people like Udell stand out. Thanks, as always, for the great episodes. Mark Stenglein is an assistant professor at Colorado State. I love viruses.
No.
Not other
I'm
aspects.
shocked.
I like just virus.
I am
just virus.
absolutely shocked over it.
All right, so I sent this to John Udell, and he had a response. Thanks for the feedback, which addresses a critical question. What is a reasonable effort for a young scientist? I love that you actually looked at the data. This is written to Mark, the data in my plot, and I admit the plot is somewhat inaccurate at the 40-hour mark based on my experience, depending, of course, on the scope of the paper and how much the data the first author personally generates. I am assuming nearly all of it. Probably the output at 40 hours a week would be one paper every 3 to 4 years, while at 80 hours a week, the output would be 1 to 1.5 papers a year. So a twofold increase in work gives a fourfold or greater increase in productivity. This is a rule of thumb, of course, and is based on the average individual postdocs in my case, since I've had very few students in the lab. I've certainly seen individuals who are fully competitive for PI positions working 40 hours a week. This requires smart planning and iron discipline. Even with this, I would guess that work time is typically supplemented by an hour or two a day at home, planning experiments, analyzing data, writing papers, and reading the literature, adding approximately 10 hours to the actual work week. You are absolutely correct that I don't have data to support this conclusion, but would be very interested in the opinions of other PIs who have been around the block. I would argue that the data cited in The Economist, which deals with factory work, does not apply well to a creative and competitive field like biomedical research. First, the competitive nature of science means that there's a good chance that one or more other labs are working on a similar project. We all know the costs of being scooped. Depending on the exact project, the impact of the paper can drop considerably. Even if the efficiency of the low-hour postdoc is higher, a la The Economist, they will still get to the finish line after the high-hour postdoc. Second, the high-hour postdoc is likely to be more driven than the low-hour postdoc. A critical part of solving difficult problems is obsession, which is usually essential to discovering something fundamentally new and important. And with obsession comes working long days and weekends. There's a fantastic talk from Richard Hamming that I came across in combing the internet for career advice. It gives a link to that. Even though it is addressed to mathematicians, physicists, engineers, it applies to all creative and competitive fields. I urge TWiV listeners to read the entire essay, which I would describe as distilled wisdom. Here's 2 highly relevant parts for the present discussion. All right, so 1, knowledge and productivity are like compound interest. Given 2 people of approximately the same ability and one person who works 10% more than the other, the latter will more than twice outproduce the former. The more you know, the more you learn, the more you learn, the more you can do. The more you can do, the more opportunity. It is very much like compound interest. I don't want to give you a rate, but it is a very high rate. Given 2 people with exactly the same ability, the one person who manages day in and day out to get in 1 more hour of thinking will be tremendously more productive over a lifetime. I took Bode's remark to heart. I spent a good deal more of my time for some years trying to work a bit harder, and I found, in fact, I could get more work done. I don't like to say it in front of my wife, but I did sort of neglect her sometimes. I needed to study. You have to neglect things if you intend to get what you want done. There's no question about this. 2, now, again, emotional commitment is not enough. It is a necessary condition, apparently, and I think I can tell you the reason why. Everybody who has studied creativity is driven finally to saying creativity comes out of your subconscious. Somehow, suddenly, there it is. It just appears. Well, we know very little about the subconscious, but one thing you are pretty well aware of is that your dreams also come out of your subconscious. And you're aware your dreams are, to a fair extent, a reworking of the experiences of the day. If you're deeply immersed and committed to a topic, day after day after day, your subconscious has nothing to do but work on your problem. And so you wake up one morning or on some afternoon, and there's the answer. If my books, essays, or talks come across as bragging about my effort, this is about the farthest thing from my mind. I really am just trying to provide advice to help young scientists and have no other agenda that I know of anyway. Who's to say what lurks in the unconscious? I have received feedback on the career talk from dozens of investigators around the world. The one thing they universally agree with is that many of the young scientists they see lack the drive needed for success. This might always have been the case. The song "What's the Matter with Kids Today" from Bye Bye Birdie comes to mind. It ends, "Why can't they be like we were, perfect in every way?" But it doesn't mean it's wrong. Finally, I want to emphasize that a career in science will be a lot easier and more fun if it doesn't seem like work. While every minute will not be joy-filled, scientists should generally feel lucky to have the opportunity to spend their lives doing something so cool. This makes up for a lot of the angst and uncertainty. That being said, it is still incumbent on older scientists to improve the working conditions for younger scientists, not just compensation issues, but the whole system of funding research, publishing papers, and the career ladder.
That's a great letter.
It's really good, really thoughtful.
So, I have another thing to add to this, and that is that if you have a passion for something, You're always working. There is never a time in your awake day that you're not working. You're driving home from the lab and you're thinking about what you did. You're at dinner, you're thinking what you did. You go to bed, you can't get to sleep because you're still thinking about what you did. The passion drives you to the solution. So how do you measure that in terms of input hours? That's not 40 hours a week. That's like 120 hours a week because it's consuming your thoughts. And it's absolutely true. You tend to neglect the things that are petty in your view, like doing the dishes, taking out the garbage, taking in a movie with your wife, taking care of the kids, driving them to soccer. The whole time your mind is absolutely trying to figure out what the hell is it like to be trichinella inside of a nerve cell, inside of a muscle, and how can I best do an experiment that will reveal the life of that organism. I spent all of my waking days consumed with that thought. And I agree that it's something that people, you can be judged by this. And, you know, how many hours a week do you work? Remember, you had to fill out the work forms at Columbia. They wanted to know how many hours a week did we work? Are you kidding me? Come on. How many hours are there in a week? You know, it's crazy to try to put this into words because can you tell me how many days you went home from the lab, Vincent, with a virus problem on your mind? Like the pH must be wrong, or I've got the wrong cell type, or I've got, what the hell is the receptor for this thing anyway? It's got to be this, it's got to be that. And you're plotting experiments. You wake up at 3 a.m, you can't get back to sleep. You write it down, you go into work, bam, that was it. There's no greater feeling in the world than to go into the lab the next day when you're struggling and have the discovery waiting for you when you get there. You can see a plaque assay, you can see a gel, you can see a cut section. I think it's fabulous to be a scientist. It's the best thing in the world.
But
so this is
what you're saying though. I mean, you point out when you go back into the lab the next day, implying that you actually left.
Well,
and I think that's a really I think that's a really critical distinction that John's letter is kind of missing, where he's talking about the importance of being obsessed. Well, yes, when you get tucked into a problem and you're focused on it and it's occupying your waking hours, that's part of science. That's normal. But what I got from the work hours plot, which I similarly noticed had, you know, a lack of data behind it, was this notion that you're supposed to be in the lab working.
No, that's not.
And I think that's what Mark is objecting to, is the idea that you'll actually accomplish twice as much by spending 80 hours a week in the lab. And I would lean toward Mark's position, which at least for some people, maybe even for a lot of people, that's not going to be the case.
Right.
That you're not going to steadily get more done with more time in the lab.
So I'm really, I agree with you, Alan, and I'm really passionate about this. This has been a lifelong issue for me. So John says he'd be interested in the opinions of other PIs who've been around the block. So I'd like to relate my experience in this regard, tempered by the fact that my career, if you start from when I entered graduate school, goes from 1970 to 2015, so it's a generation ago. However, it was made very clear to me explicitly when I entered graduate school, this same thing, that science required a particular type of dedication that manifested as spending a lot of hours in the lab, and it seemed that you got bonus points for being there at night and on the weekends. And I saw, it drives me crazy that people are judged by whether they're there at weird hours. Okay, nights and weekends are a big deal. And this to me was shocking, and it ran totally counter to the way I wanted to live, and so I didn't. Okay. I worked somewhere between a 40 and a 50-hour week. Okay, at my peak, I was in the lab working at 7:30 in the morning, and I went home about 5 or 5:30. I saw my kids in the morning before they went to school, and I was home for dinner every night. When I had to, because an experiment called for it, or I had to prepare a lecture or something, yeah, sure, I was there Some nights go back in to start an overnight, sometimes a weekend preparing a lecture or something like that. But basically it was 40 to 50 hour weeks. And I have to say also for the young folks out there, I never had a big lab. It was probably never more than 10 people, including technicians and undergraduates, probably more like on average 6 people at any given time. 2 papers a year over my entire career. That totals less than 100 papers. No cell papers, no science papers, no nature papers. Okay. I had 2 PNAS papers, but I didn't have any clunkers. They were all either, the vast majority were either first author or, last author publications, all in good, respectable journals. No headlines, no big deal. I never gave an ASV plenary talk, but I made contributions. Okay. And I'm particularly proud of the Fields chapter. I'm very proud of participating in TWiV. I educated a lot of students. I was a university professor. Okay. I had a perfectly reasonable career. All right. And, if I had worked harder, I might have accomplished more professionally. Okay. And most of the people I know who are superstars professionally work very, very hard. There is that kind of correlation. But I don't think it's the case, at least it wasn't when I was doing it, that you absolutely have to do that in order to succeed. And I'm concerned that this idea that has to be the case is going to turn away people who want to do science. All right. Now, you could argue that if you want to do science, you're going to do it. All right. And I think there's a certain amount of truth in that. But I was also extraordinarily efficient. Okay. When I got into the lab, I knew exactly what I was doing. And it was a game to me to multitask in a fashion that maximized my use of time. And, nowadays, if I were doing it, I wouldn't even turn on my computer, all right, until I had the experiments that were going to require the most time working. And I would never, ever, ever do anything in the laboratory, in particular on my computer, that wasn't related to work. Okay? All right? No Facebook.
Nobody's like you anymore, man.
How the hell did you get picks of the week?
No, he's talking about before. Nobody's like you anymore, Rich.
So my point is it can be done. It can be done. You don't have to.
You have an older view. How do you know today it could be done that way?
I don't know. And that's why I want to hear from other scientists. I think there are plenty out there who can do this. So they need to write in.
Yeah, sure.
So we were talking about this earlier, and let's hear from them.
I don't think it matters how long you work. I think what matters is what you produce, what you publish. I've seen people work 40-hour weeks and publish great papers, and people, if you need to work longer, that's fine. To think that there's one rule that fits all is kind of silly, right?
I was there.
I think just whatever it takes for you to get your papers done.
I was at Rockefeller for 3 years.
Maybe you suck at pipetting and you need 80 hours. Who knows?
I was at the ivory tower of ivory towers, right? I observed behavior of all these people that you could observe. And most of them ended up at the faculty club around 5:30 or 6 o'clock. And they were there until maybe time for dinner. And then they went home and then they came back to work at 9 o'clock. And they were not ordinary people, but these were people that they were aware of the fact that they had a life in addition to that. And there were concerts and there were art shows and all kinds of other nice things to distract you from your science so that you could take a breather every now and then, so that you could relax, so that you could maybe come up with a solution that was bothering you, that because you pressed on it and, you won't get anywhere like that. You have to have some downtime. So, and everybody, how about you, Kathy? How do you feel about all this? Because you're active.
So I do want to have some comments for Mark and for some of the listeners. 2 things. Rich's philosophy was espoused in a way by a pick that he made on TWiV 243 at Penn State. It was a Scientific American blog, and it was the awesomest 7-year postdoc, or how I learned to stop worrying and love the tenure track faculty life. And the 7 points that this person made, one of them included, let's see, I have to scroll back down to it. Sorry. I worked fixed hours and in fixed amounts. I tried to be the best whole person I can. Mm-hmm. I have fun now. So that was written in 2013, so pretty contemporary. And then also, I want to point people to a blog post by Megan Duffy, who's a faculty member in ecology here at University of Michigan, who tweets a lot, but also wrote an article entitled, "You do not need to work 80 hours a week to succeed in academia." And she cites exactly what the concern was that we heard just a minute ago. I hear routinely from younger folk, often women, who are seriously considering leaving academia primarily because they think that a tenure-track position would require working so much that they wouldn't be able to have any life outside work, including raising a family. So this is my attempt at slaying the zombie idea that succeeding in academia requires working as much as an investment banker. And then I think then she's got 2 asterisks to that. And as it turns out, investment bankers are being encouraged to work less.
I think making general statements, though, is wrong. Because it may be at some point in your career, you need to put in an 80-hour week to get something done.
Oh, right? Special times
to say you never have to do
activities. That's true.
Well, no, but this is a discussion about, in general, should you expect to have to work 80-hour weeks? And I think in general, that is not a realistic expectation to have or to promulgate,
I think 80.
Particularly in a complete and utter absence of data. Wow. And I mean, I've got to pick on the response that John gave here, where he's throwing out numbers that don't come from anywhere.
Look, as I said, I think what matters is what you can do in the time that you can put in, right? And nothing to say you need to do 40, 50, 60, 70 is kind of ridiculous. So,
and it's also not true. It's also not true. Somebody might say, "Oh, well, but if you're that efficient and you can get your work done in 40 hours, just imagine how much more you could do in 80." But that's not.
No, that's not true. Right?
It does not scale that way. I'm really convinced that it doesn't scale that way, and I don't think that creative work is going to be more responsive to that than factory work. I think to a large extent it'll be less responsive because as you get fatigued, you get less creative.
This is true. Very true. Every now and then you screw yourself up with an experiment that takes 3 days and you've got to take samples every 4 hours.
Of course. Yeah.
And that's absurd. It happened to me once on Christmas of all times, but otherwise, no, you don't get embroiled in stuff like this. What about field ecologists? For Christ's sakes, they spend all of their time in the field. They're working virtually every moment of every day they're out there. So, you can get overworked, you can get burned out, you can get tired. And you're right. You're not accomplishing anything after wherever you reach that point, right?
I'd be interested to hear from others about their careers. Let's hear your stories about what kind of hours you put in. What do you think is important? Yeah. All right, let's do some picks. Alan, what do you have for us?
Okay, this is totally not a science pick, but I couldn't resist. This will be, it's Thanksgiving weekend, and so today, of course, is Black Friday. It's Christmas shopping season, and if people are looking for, they've got $600,000 burning a hole in their pocket, and they're looking to buy something special for the TWiV crew. This is Sail Magazine's Best Boats of 2018, and I've highlighted their a particular choice. And if you watch the video and you're a regular TWiV listener, you will probably have a very disorienting experience, and that's why I'm picking it.
I haven't watched the video yet.
You should go ahead and watch the video, and I think you'll get what's going on.
It's this thing that starts with the gas burners in front of it. That video.
Yes, that is the video.
It's got sound.
Oh yes.
Boom.
So that video that starts, that's actually the galley of this luxury catamaran. And there's a guy who's going to walk around and give you a little tour of the features of this boat.
Have you ever been on one of these boats, Alan?
Oh yeah, I've been on a bunch of them. You'll watch the video and you'll see.
Okay.
You'll see why I might have had some experience with this type of world.
All right.
Rich Condit.
Walter Isaacson's latest biography, Leonardo da Vinci.
I heard this.
I actually have an autographed copy.
Wow!
Why is that?
Because in Austin they have so many cool events here. They have a book festival that they close off Congress Avenue and they have authors come and give talks and stuff, and lots of vendors and publishers and stuff. And Walter Isaacson attended this, and he actually gave a talk. They used the Capitol building, probably the best use of the Capitol building. They used the Capitol building to give talks. So Walter Isaacson gave a talk in the Senate chambers.
He's cool.
So we went to the Senate chambers and heard him talk. And by then I was about a third of the way through the book already. I took the book with me, and he had a signing afterwards, and he signed my book. And it's great. It is absolutely terrific. It gives, I had not much insight into Leonardo. Actually, Leonardo
Speaking
was really
workaholics.
Well, yes and no. He finished very little.
Oh, true.
Right.
He didn't publish.
Oh, yeah, ADDH to the nth.
Yeah, he kept a lot of notes and stuff, but he hardly finished anything, even his paid commissions. Okay, right. Very, very interesting. The Mona Lisa he was working on for about 13 years. Hey. He carried it around with him, and I don't think he would have kept working on it if he hadn't died.
What about this latest painting that was sold for
$450 million?
Yeah, he doesn't get any of that.
No royalties, nothing. Damn.
Actually, one of the interesting things about that's discussed in the book. Okay, so this is Salvator Mundo, right?
Yes,
it's a picture of Jesus
with a
holding, giving a sort of with
orb.
one hand held in blessing and the other holding this crystal orb.
Exactly.
Okay.
Exactly.
If you look at the crystal orb, that's not how it should be. If you look through a crystal orb, the image on the what's on the other side will be upside down.
Right.
Okay. Mm-hmm. And Da Vinci knew that. So the question is, why did he not? Because he was a stickler for details. Why did he not do that in his painting?
Well, the only person that could possibly reverse the image was Jesus.
That's one of the possible explanations. Okay.
Rich, what did you like about this biography?
I knew very little about Da Vinci. Okay. Except, just sort of the casual, he was the Renaissance man and, thought about just about everything. And this just, put the whole thing together. It was a very thorough, complete biography of Da Vinci. And I think probably the most interesting thing is that, as I said, he was obsessive. Okay, he was absolutely fascinated with everything in particular in nature, and translated what he learned from nature into his paintings. I mean, the obvious is that he studied anatomy in order to inform his paintings of the human form. But I was fascinated by the fact that he never finished anything.
So don't be impressed with everything he did, by the way. He got the circulation room.
Yeah, got circulation.
His helicopter never would have flown either.
No, but a lot of other things did work that they've tried out. So this is fantastic.
Most of his day job mostly was organizing festivals and shows for the royalty in the various towns where he lived. Okay, he was on staff to do that kind of stuff. That's where he made most of his money.
And he was obsessed with warfare.
That's kind of how he sold himself. I was interested to understand whether or not he was in fact famous and a legend in his own time, and he was.
Yeah.
Okay.
Yeah. Someone once commissioned him to do a painting. I heard this through somebody else's telling stories about him. And they wanted an example of his art. So he said, get me a piece of paper. So they brought a piece of paper over to him, and he drew a freehand circle that was perfect. Just bring this back. Yeah. There was
I've heard that, yeah.
It was absolutely spot on. So we had the privilege of going to the museum in Florence and seeing all of his inventions actually, built. It was amazing, actually. My wife is obsessed with his painting as well. Yeah, as well she should be. It's an amazing deal.
Dickson, what do you have for us?
I hope I haven't picked this before because I found it in my picks of the week and I didn't see it had been picked. It's the latest winners of the Nikon Small World Contest, and lots of great stuff.
Are you one of them?
They're beautiful.
No, none of them.
They're beautiful.
My nurse cell was the winner in the 1976 contest, by the way.
Oh,
I told you that before, though.
Yeah, no.
yeah.
I didn't take the picture. Somebody else
How
did.
many hours did you work on that?
Not enough, maybe.
Skip ahead to number 18, which is skin. And it's the most amazing picture.
Oh, you like this? Oh, I'm glad you all like this.
Yes.
I love these things because seeing is believing.
Up at the top is where you can scroll through them all. I've been doing it
good.
periodically.
Good.
Yeah,
Good.
I love it. Yeah.
Good, Kathy. I'm glad you
Great.
like this.
God,
The
they're
first
gorgeous.
picture is just amazing, though, isn't it? I mean, it's just, it's amazing.
And number 18, though, is this polarized light, all these anchors,
Yes.
and it's skin.
It's skin of a sea cucumber.
Yeah.
That's amazing. Look at that.
It's just bizarre.
Whoa.
Yeah, life is, it's so involved. It's so beautifully
When
involved.
you said 18, and so I looked at it and I saw it and I said, "Oh, this is some
Anchors.
kind of joke."
Yeah.
They don't have the picture, so they put this in the background. So that's for real, huh?
It's
Yeah.
Holy
Apparently.
cow!
Yeah,
That's pretty cool.
quite amazing.
Kathy,
Right.
what
Older.
do you have, Kathy?
I picked something that Vincent and I got when we did the TWiV at Indiana University. We got these 2 capsids that are self-assembling. They were printed on a 3D printer, and they have magnets to hold the capsomeres together. And I have accomplished it twice. If you break it apart completely into pieces, one of them, and put it in the box that they gave us, which is a good size, and shake it, you can get it to self-assemble. And it's just the coolest thing. It's really, really neat. And then, so I wrote back and got more information about it. So Jolene presented us with these, and Thule worked on it, and other people there at IU did too. They have a video that they made about patterns that goes along with this exercise. So they have these capsids that they use in an outreach program at the Wonder Lab, which is a children's science type museum in Bloomington, from what I could tell from the website. So there's, I hope Vincent will somehow put the picture in of ours, and then there's the video, and then there's the link to the Wonder Lab. So
This video, at one point, they have a big jar full of capsid subunits, more than one, and they are all they don't show the whole thing, but many of them self-assemble. It's really During the video, during our TWiV, I was shaking it and nothing was assembling, and Adam said, "You're shaking it too hard. You can't assemble under those conditions."
Hey, someone should come up with, get this one now, this concept for the 3 segments of influenza virus. And so you could study recombination this way.
Reassortment.
Yeah, reassortment. I meant reassortment.
Yeah, sure. We could do that.
Would be
Make
very
metamagnets, Dickson.
Yeah, okay.
You get one long RNA.
Here we go.
All right. My pick is a Times article from November 17th entitled, "Science is dry, obscure, complex. Well, it makes for great comedy." And this is an article which is tracing how people are using comedy to teach people science. There's a lot going on with that, particularly here in New York. New comedy podcasts, live shows, things like monotony, drunk science, drunk education, right? And there's Ben Lilly, who is here in New York City, used to work for TED. He's one of the people involved in this. He's trying to make he does a podcast called Story Collider, where he has scientists tell their funny stories for 10 minutes at a public space and so forth. Comedians are getting involved. And so it's interesting. I mean, I asked someone recently, do we have to get a comedian onto TWiV, to tell jokes? And they said, well, I don't think you have to do that, but this seems to be a way.
Because we're all aspiring comedians.
We are aspiring comedians. I mean, we can be lighthearted here.
We can. Sometimes we're accused of being a little too lighthearted.
But we certainly don't tell jokes. But anyway, it's an interesting idea. It's not where I'm going to go because I'm not a comedian, but there is clearly an interest, and people want to hear science in the context of jokes, I suppose.
Yeah.
We have a listener pick from Ben. Hello, Team TWiV. I was just listening to TWiV 467, which, if it were a journal article in search of a descriptive title, might have been called John and Ted's Excellent Discussion of the Problems and Inequities in Scientific Research. This discussion inspired me to send you this listener's pick. And he sent a link to an article in The Guardian. Is the staggeringly profitable business of scientific publishing bad for science? It's a bit of a romp through the troubling history of scientific publishing. I would love to hear the thoughts of the TWiV team on this article, which I think covers some new territory on a subject that you all have discussed many times. This has been brought up before, Alan. I think it has been here.
I think it has been, and I don't know if I picked it or if I meant to pick it, or it's certainly something we have discussed, but this is a very, very good deep dive into where our scientific publishing world as it now stands came from, and what that has led to.
So Elsevier is one of the focuses of this, which has global revenues of $19 billion.
Why would they ever want to give that up?
It is 36% margin, higher than Apple, Google, or Amazon in 2010.
Yep. Very
profitable. Yes, and that's why the original Cell has become Cell Host Microbe, Cell Immunology, blah, 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 et cetera, all exclusive. And as we've talked about many times, this is screwing up science because now you can't get a job at many places unless you have a Cell Science or Nature paper. So people outside of your immediate circles are making jobs, and I don't think that's right, but you can have your own idea about it.
There's an author revolt going on right now.
Not much, though.
200. I read 200 up to the last.
What did you read?
I read that 200 separate authors or scientists in various places refused to publish in any of those journals anymore.
It's not enough anymore.
No kidding.
Well, we've talked about 200,000. It's a good article because, yes, as Alan said, you learn how we've gotten to this point. And boy, it's really profitable. That's why there are all these weird journals popping up who want to grab your paper and charge you to publish.
I have a question here that relates very much to the way articles are accepted for journals, because, you get a lot of rejections from science saying, this is a great article, this is fabulous work and everything else, but it's just not so appropriate for science. You look through science and it's got articles on nanoparticle sublimation into the presence of 15 different ions. Come on, give me a break here. That article appeals to 3 other people. And, and I'm not kidding. They really do. And I read these articles at home to my wife. I just read the titles, and the titles have unpronounceable scientific terminology in them for the most part from these, the material sciences groups. And so those are poor man's excuses for rejection. That's not the real reason that article was rejected from Science. What is the real reason? And that is you didn't have an advocate that was recognizable at the editorial level. So the editorial boards for these journals are the ones that really do determine who gets published and who doesn't. And those are clubs, right? You're saying those are clubs.
That's part of it. It's not just that, though.
But there's a lot of that.
There is a lot of that. But the journals are also under tremendous internal and external pressure to focus on high-profile papers that are likely to get a lot of attention.
The next Watson and Crick.
And so you are looking at material science papers with unpronounceable titles, but they actually could be interesting to a much wider swath. If well explained, and could have a significant impact that'll be well covered. And by the way, the material scientists look at the molecular biology and the parasitology papers, and they say exactly the same things.
Of course. So that's not the reason why they get.
And why do these immunologists have different labels for all the same things?
Exactly.
So it's about the perceived impact of the paper and the perceived public relations splash that the paper will make and what it will accomplish for the journal.
So, Alan, who makes that decision?
It is generally being made at the level of the research editors who are triaging the papers.
Sure.
And they're former graduate students at postdoc went into the publishing business, and it's a darn good job. You get to, you read science papers for a living.
Yeah.
Now, the bad part of the job, of course, is that 99% of those you're sending a reject notice to, and then they harass the hell out of you. But that's, that goes with the territory. And so these people are supposed to be very involved, very immersed in the fields that they handle papers for, and they generally are, and they know who's who, and they know what's going on in the field and what's likely to make a big impact. The problem, as I see it, is that there is this tremendous pressure for them to focus on high impact papers that are likely to make a splash, and it skews the entire literature,
Sure. Yep. It determines who NIH funds also.
And who gets jobs.
And who gets jobs. And then it's self-fulfilling.
Problem.
Self-fulfilling.
All right. That's TWiV 469. Apple Podcast, microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. Send us your cash, microbe.tv/contribute.
Sound just like Elsevier now.
Publish in society journals and send us your alternative cash. No, seriously, if you like what we do, you can help us out financially. Microbe.tv/contribute. Dickson Despommier is at thelivingriver.org and what's that? Parasiteswithoutborders.com. Dickson, thank you very much.
What an enjoyable day after Thanksgiving. This was great. I really had a good time.
I could spend 80 hours a day doing this.
I could do this. We would burn out. I know we would be like slurring our R's or something at the end of this, past the scotch or whatever.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. I agree with Dickson. This is a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. I think I'm going to go outside.
Do you miss the 40? Do you miss the 40 hours at your bench, Rich?
I always enjoyed the time at the bench. There wasn't enough time for that.
Your voice tells everything. That's absolutely right.
Alan Dove is at alandove.com, turbidplaque.com, Twitter, Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure, though not one that I would want to last for 80 hours a week.
You wouldn't want to do TWiV 80 hours a week? No.
No, that would be too much.
That's too much. You need to do other things. I totally agree. I think once a week is good. However, I could do a podcast a day, but they'd be all different.
You almost do.
You do a podcast. What are you talking about? I could do a podcast.
But I like chatting. It's fun. And more so, I like science, but not all science, because I don't know anything about materials science, as you were just talking about.
Well, neither.
But the things, and then I can ask you questions about plants and
you could give you all the wrong answers, and our listeners will send in the right answers, and we're all off and running.
By the way, we did TWiVo this week, and Nels's pick was a video of a fungus killing a worm. They wrapped these rings around them and choked. Oh my gosh, I felt so badly for those worms.
They don't eat live worms, so they have to kill them first, and then they eat them.
Then they put these they assemble these things on their length, and they pull things out, and then these new spores are formed or whatever, and they're shooting at it. Oh,
they're
wow.
Fantastic.
Amazing.
They are good.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiV and the sponsors of this episode. The Department of Microbiology at the Icahn School of Medicine at Mount Sinai in New York City, and the Virology and Gene Therapy PhD program at the Mayo Clinic Graduate School of Biomedical Sciences in Rochester, Minnesota. Remember, if you're interested in those programs, December 1st deadline. Introductory music on TWiV by Ronald Jenkins, ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.